About biotechnology application in medicine, right? So the recombinant DNA technology process have made immense impact. So what is recombinant DNA technology? So none other than we are taking the gene of interest and we are inserting to the plasmid, right? After inserting to the plasmid, now this gene we call it as recombinant DNA. Now this recombinant DNA you can insert into bacteria, plant cell, animal cell or fungi. So now let's see how this recombinant is very helpful for genetically engineered insulin. So in the area of healthcare, by enabling the mass production of safe and more effective therapeutic drugs. Further, recombinant therapeutics do not induce unwanted immunological responses. Assume that we need insulin. That insulin can be taken from pig, cow or whatever the thing. But that insulin will immediately causes the reaction. In order to prevent from that reactions, what we are doing in case of similar products isolated from non-human resources, we get allergic infections. So we are taking or we are extracting the human gene which produces insulin or which produces certain enzyme, and we are inserting to the plasmid, and that plasmid we are moving into the plant, animal, or at present about 30 recombinant therapeutics have been approved for human use in the world over. In India, 12 are presently being marketed. At present, about 30 recombinant, you have to remember this number, in the world 30 recombinant therapeutics have been approved for human use the world over, and in India 12 of these are presently being marketed. So genetically engineered insulin. So what is insulin? Insulin is a hormone which is produced by beta cells of islets of Langerhans. And so this hormone, what it causes? This is a peptide hormone, polypeptide hormone. So let's talk about this. The management of adult onset diabetes is possible by taking insulin. Usually they have to take insulin at regular time intervals. So just before, half an hour before consumption of meal, they have to take the insulin. So what would be the diabetic patients do if enough human insulin was not available? You have to take a supplement of insulin. So if you discuss this, you would soon realize that one should have to isolate and use the insulin from other animals. Would the insulin isolated from other animals just be as effective as the secreted by the human body? No, that will cause immediately reaction, allergic reaction. Our body will become hypersensitive. And would it not elicit an immune response in the human body? It will not elicit an immune response. In our body, question mark, means it will elicit an immune response in our body. Now imagine if bacterium were available that could make human insulin. Right. Suddenly the whole process became so simple. You can easily grow a large quantity of bacteria and make as much as insulin as you need. Think about whether the insulin can be orally administered to diabetic people or not. No. Because insulin is polypeptide. It will be digested once it is taken through oral. It has to be injected through the IV route only. So this is insulin. In the insulin, we have, so in this diagram, you can able to see the proinsulin. Here we have the disulfide bonds. The same disulfide bonds will be seen in immunoglobulins as well. So between lightweight chains and heavy chains, between light chains and heavy chains, and between the heavy chains we can able to see the disulfide bonds. The same disulfide bonds you will be seeing in insulin. So here this is the proinsulin, which is inactive form, and whatever we are seeing is the activated form. So we have A, blue is A and green is B, and this one is the C, right? So A and B, A peptide and B peptide. So this both are linked by disulfide. In the proinsulin we have the C polypeptide chain. Now after we have to free this C polypeptide chain, then this will be the active insulin. This is inactive insulin. This is inactive. When C polypeptide chain is attached to the A and B, between A and B, then this is inactive. When this C is separated, then that will become active. So insulin for diabetes was early extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source though caused some patients to develop allergy or other types of reactions to the foreign protein. So insulin consists of two short polypeptide chains, chain A and chain B. They are linked together by disulfide bridges. In animal, including the humans, insulin are synthesized as prohormone, like a proenzyme. The prohormone also need to be processed before it becomes fully mature and functional hormone. Which contains an extra stretch called C peptide. The C peptide is not present in the mature insulin and it is removed during maturation into insulin. The main challenge for the production of insulin is during rDNA, which recombinant DNA techniques, was getting insulin assembled into mature form. In 1983, Eli Lilly. So remember this, 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin. So introduced them in the plasmids of E. coli to produce the insulin chains. So chain A and chain B were produced separately, extracted and combined by creating disulfide bonds to form an human insulin. This is very simple. But remember this year, 1983, the company name is Eli Lilly, American company. This is an American company. They prepared they extracted two separate genes for polypeptide A and polypeptide B. They extracted, so now the bacteria has produced separately, E. coli. Now they both joined. So this is the very easy point. So when it comes to insulin, so this is genetically engineered insulin.